Hi, this is Miss Litton, and this is 7th period support for AP Biology. Say hi.
Hi.
Okay, and this is concerning our Unit 7 exam. A couple things I want to go over with you. When I wrote the test, the multiple choice, I literally went down and picked 5 to 6 questions off of each one of these chapters. The only time I took one question was, I think, senses. Just because we went over that really briefly. Remember, that was just like the type of receptors and understanding signal transduction was the limit of that. On your essays, I went through for each of your essays, and I wrote them side by side, your 2 different class periods, and I went through and picked an excretory question. I went through and picked a nervous system question. I doubled down on one of them for each chapter, for each of your tests, because I didn't ask a complete essay on this one. Went through and hit every one of these. Now, where did those essay questions come from? If you go to the essay review, it could have come from that. It could have come from an application out of that, or just general content, you know, but by preparing for those essays, it puts you in a good spot. The other thing I want you to know is yesterday afternoon, I think by 4 o'clock, I had entered all your grades, makeups or anything that you had done or anything that needed to be adjusted. The only thing I didn't put in there was your Google form from yesterday. But otherwise, everything else is in, so you can go and make your grade comparisons. As far as what's going to be acceptable, remember you want to be within 10% of each other. If you are 10.4% away from each other, I will be okay with that, so I don't want you to stress about the tenths or things like that. But, you know, I don't want somebody who's 15%, you know, away or anything like that. If you have a concern or question about it, you can check with me. If in the morning, the sub will have my number. Some of you have my number. You can always text me. Some of you have my number, don't you? I'll give somebody my number. It doesn't, I don't care. Just, you know what? I gave it out before one time, and over the summer, I got inundated by a 9th grade, it had to be a 9th grade child, and it was super annoying. I normally would just trust my number, but they like to message me at 11:30 and 12 o'clock at night, and they would just ping me and then say really inappropriate things. So I was like,
What? Me? Wasn't me.
Was it? No. Anyway, so I'm glad we got that recorded. Super fun. All right. I spoke to a couple of you in each class, but I want to remind, just so, since there's so many of you right here, when I'm just writing to the sub that you'll know how to, I haven't written the sub plans yet, so I've got to still do that. But I'm going to say they'll know how to move when the test starts. So, yeah, so some of you I've specifically tasked with making sure we follow through with that. But, you know, sit in your regular seats for the subs so they can take roll more easily, okay? But then this will be a stay, go, stay, go, stay. Then starting right here, this is a go, stay, go, stay, go, stay, stay, go, stay, go, stay, go, right? So you're not right next to somebody. The outside ones, the outside edges are staying. You're a go, and then it alternates back. When you go to a lab bench, go to the far side of the lab bench and sit together as a team. I'm going to give you one paper. You'll have an exam, but then you will have 2 papers. I'm going to staple them together so they don't get lost. Just gently pull them apart. One of you can hold one sheet, and one of you can hold the other sheet, you know, for writing on the essays. And you each will have your own scratch paper on the other side of the paper. Okay? Questions or concerns on how the test is given or anything like that?
Oh, yeah.
Yes.
Are we going to fill out like one multiple choice sheet?
Yeah. Is that all right?
Yeah.
If you disagree, do you know what to do? Yeah, just, put your name and put what you think. Like, I think this one should be, you know, C. If you are on the essay and you disagree, use the scratch paper and just box it and put your name around it and say, please grade this, and I will. Okay? I don't want you to ever get something that you don't agree with, ever, 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 and don't ever use that as an excuse. Okay? Any other questions or concerns on the process? All right, so let's talk about each of these chapters. Are there some big ticket items that you are concerned about in the excretory system? Anything?
I hit that one
Yes.
I don't know if it's a big ticket item, but I just want to know what's the big significance about the reabsorption of salt, in like, why
Okay, so for salt, remember the reason why we would reabsorb salt, that's not really our goal so much the salt, the sodium's important. What are we really after? Water. Because by reabsorbing the salt, we create a hypertonic environment so that the water will follow it. So that's the key there. Any other issues with the excretory system? Yes.
The descending loop it occurs chronologically before the ascending loop, right?
Okay, so you'll if we're talking about the urine tube, okay, so you have your proximal. Okay, so if what I've shown you there is I haven't shown you the blood pathway, I've shown you the urine pathway, right, or the filtrate. So this right here is the what? No.
Bowman's capsule.
Yeah, the Bowman's capsule, what's another name for it? Glomerular capsule. Then the closest thing to that is the proximal convoluted tubule. Then this one goes descending, and this one goes, because that's the only path you can do. So don't do it like a memorization, do like, well, we're going down, so that has to be descending.
Can't ascend.
Yeah. And then this is your, right here, this is your distal convoluted tubule, and then this is your collecting duct. Yes?
The descending and ascending tubes, they're in the renal medulla, right?
Yeah, well, parts of it. You've got, remember you've got the cortex, the medulla, and the pelvis, so neither one. But you have it about halfway through here, but I didn't have you differentiate that, so you don't have to worry about it. Yes? Oh, sorry, wait, he was next, I'm so sorry. Yes?
Okay, the kidney filters out the urea.
I can't hear, shh, go.
The kidney filters out the urea, but what produces it?
What produces urea? The liver. The liver. Remember that picture with the fire pea that I showed made? Okay. So remember I showed you a steak going in a mouth and it getting digested and then in the stomach. And then I said the second place it goes after your digestive system is once the amino acids are absorbed, that they're going to go to your liver, and that's where deamination occurs, where you take off the amino group, the NH3. Okay, and then that combined with some carbon dioxide and another amino or a group is how we eventually form urea. Right? Okay. Other questions on the excretory system that you would like me to go over? Okay, if you think of some more, that's fine. Come back to it. Okay. Okay. Oh, okay. All right. Next, let's talk about, that's a big chapter, the nervous system. Okay. So things that you would like to discuss about the nervous system. Let's hear. Raise your hand. Raise your hand. Oh. Yes.
For the reflex arc, would we have
Yes.
to describe how like sensory receptors go through the dorsal root and like all that?
I would know that, yeah. Sure. So when you have a reflex arc, let's say you have a hand and it's touching a hot cookie pan, chocolate chips, okay? So you have a sensory neuron, right? The sensory neuron that's going to detect that heat. Here is the, what is that I just drew there? No. Cell body, right? In the dorsal root ganglion exterior to the spinal cord, right? Of course. Okay. All right. And at that time, it's then going to synapse with a what? Good. An interneuron, and that interneuron is going to go where? It's going to go to the brain. Good. And then you're also going to be synapsing with a motor neuron that's going to go to your big muscles right here to make your hand pull away. Okay, and the sensory neuron goes in dorsally to the spinal cord. The motor neuron goes out the front. The motor neuron cell body is within the spinal cord. Literally, there are 1,000 plus pictures of a reflex arc that you could pull up and just review. Yes?
Is the interneuron traveling up to the brain part of the reflex arc?
Well, it's part of the reflex in the way I would talk about it is only in addition to and saying that your awareness of it, because it's a reflex, you don't have to wait for the processing to take place in your brain before it responds, and the safety of that. I would bring it up like that. Okay, yes?
So it's, the sensory receptor, their cell body is not exterior of the spinal cord, but the motor neurons are.
Yes.
And
In the
also
gray matter.
is inside.
What?
Gray matter is inside the spinal cord.
Yeah.
Oh, I see,
There's the
And,
gray matter and the white matter in both the spinal cord and the brain.
I think you said for the reflex pathway, you don't have to go up to the brain, it's just straight from the spine
I actually just spoke about that.
Okay.
Okay. Yes.
So when the sensory neuron is synapsing with the inner neuron, right? Is that at like the spinal cord, or is that just?
Yes.
Okay, so at the spinal cord.
Yeah.
So there's like one neuron running from like
Yep.
the hand to like.
Yep.
That's pretty cool.
I know, right? Now, remember, in your motor neurons, in the autonomic nervous system, that's the place where you have two motor neurons. Remember that? In your autonomic nervous system, remember there's central nervous system, right? Let's just break that down a little bit. Okay. Why is that tiny? Okay. If you take your nervous system, right, you break it down, you have the central nervous system, and that consists of what? Brain and spinal cord. Good. And then you have your, what's the other one? Peripheral nervous system, and then that consists of what?
Autonomic.
Yep, auto and somatic. And what is somatic about?
Choice.
Choice. That was the word I was looking for, right? Except in a reflex, right? Because I'm not like, shall I leave my hand? If I purposely go, I am touching this hot pan now, can I hold my hand on there?
Yes.
Yeah, I chose to put my hand on there and I can keep it. Okay, but if I am unaware of the hot pan and I touch it, like the fajita pan at dinner the other night, which I forgot was in the oven, so I went to move it because it was getting too close to the guacamole, and I was afraid it would wreck the guacamole, so I go and just grab it, okay? They brought it out sizzling, everything, but I thought, I'll just move it, you know? And I go, and the boyfriend goes, what are you? And it, I was like, you know, and I'm like, that is awesome. So I got to eat the rest of the meal with one hand holding my soda, and then eating with my right hand. It was awesome. But anyway, I was unaware that that was hot at that moment, and so I didn't even think about that, but it was still using part of what you would consider, your skeletal muscles, right? So usually this is about skeletal muscles, but I don't choose that. Autonomic, what kind of muscles am I, this is automatic, or what would be another way you could think?
Automated.
Sure.
Autonomous. That's the opposite.
Involuntary, what muscles am I using?
Sympathetic brain.
No.
Smooth muscles.
Smooth? Where might I find a smooth muscle? In an organ, right? A gland. What else am I using?
Cardiac.
Cardiac. Perfect. Good. Now, you can break down autonomic into what? Sympathetic and parasympathetic, right? Which one is about chillaxing? Good. And this is stress. Yeah, fight or flight. Now, good. If it's immediate stress, what gland is involved? What part of that gland? Immediate stress. Adrenal medulla. Long-term stress over the course of this semester would be your adrenal cortex. Good, I was just checking. Okay? In your autonomic nervous system, okay, you have two motor neurons to the target. In your somatic nervous system, you have one motor neuron to the target. Okay, back here. Any more things on nervous system you want to talk about? Yes. Okay, so there are three types. Make sure you differentiate between neuron and nerve. Okay? So there's three types of, guys, I can't talk over the top of you, I'm sorry. You have to just put it in your group show notes. There are three types of neurons. What are the three types of neurons? Sensory, motor, and interneurons. Where do you find interneurons? Brain and spinal cord. Okay. Now nerves are different. Okay. When you talk about nerves, you could say it's sensory nerve, or it is a motor nerve, or it is a mixed nerve. But a nerve is the part, it's the long axon, the away part of the neuron, all bundled together. That's what a nerve is. So some of them are just completely sensory neurons, some of them are completely motor neurons, and then some are mixed. Now all the ones coming off your spinal cord, those 31 pairs are all mixed. Okay? The ones coming, the cranial, 12 cranial pairs, some are sensory, some are motor, some are mixed. Good. Okay? More things in the nervous system that you would like me to review. Okay, and if you think of something, write it down. I'll just move on to the other one. Endocrine system. What are some things you would like me to review for you in the endocrine system? Yes, sir.
Should we know, actually, never mind.
Yeah, that was a good idea to retract that. Yes.
Would a potential essay be like the thyroid hormones with like thyroid releasing hormones, stimulating hormones, and the actual thyroid hormones?
Yeah, because what might that show?
Negative feedback.
Yeah. Is that one of the, what is negative feedback an example of?
Homeostasis.
Good. Ways to maintain homeostasis. What other ways do we use?
Positive feedback.
What other ways do we use? Something, another way. Hi, Weston. What other way?
Antagonistic.
I'm not hearing it, but contrary or antagonistic
Yeah.
hormones?
Oh, sure.
Okay. All right.
Yeah, those.
Okay, so yeah, that would be something to know. What else?
Can you go to the
Yes.
control for the anterior pituitary, like not the posterior, but the
Anterior, sure. So for your anterior pituitary, it is not directly connected. So if I did the stalk like this, and I put the posterior back here, and I put the anterior again with this drawing. Yeah. Okay. Whatever. Okay, out at the front. Okay, the anterior pituitary did not descend out of the brain, okay, but the posterior pituitary did, so it's directly connected. The anterior pituitary came later, and so as a result, the neurosecretory cells in the hypothalamus only extend as far as the stalk. And there, which this is leprechaun blood, so it's green, there it reaches as far as the first set of capillaries in the stalk. And what the hypothalamus will put in there are releasing hormones. So those releasing hormones get put in, they get secreted here by the neurosecretory cells of the hypothalamus. They get put in the capillaries right here of the stalk. Now, those capillaries, remember this is where you have, leprechaun blood. Those capillaries are going to come back together and then reform a second set of capillaries in the anterior pituitary. So the releasing hormone, when it's put in, will travel then down here and come out at the second set of capillaries. These releasing hormones will tell the anterior pituitary what hormones to put back into the blood here. Okay, and what choices does it have to put back into the blood? What are the ones it can, what can the anterior pituitary choose from? FSH, what's one I could group with that? FSH and LH, which are going to impact your gonads. What's another one? Prolactin. And those are going to impact breasts. And for what purposes? Not releasing. Yeah, making or producing would be a better word. Producing milk. Good. What's another one? TSH. What is TSH going to affect? It is thyroid stimulating hormone, but what is it going to affect? Thyroid gland, so that the thyroid gland will produce what? Yeah, good. Okay, I don't need to write that. What's another one? Growth hormone, its target is what? Muscles and bones, so that they grow. 1, 2, 3, 4, 5. What else? Which affects the adrenal cortex. What else? 5, 6. I didn't give you the 7th one, so you're good. Does that answer your question?
I just want to make sure.
Yeah. Okay, yes. I can't hear. Shh.
How is the parathyroid stimulated?
Okay, so you will have feedback mechanisms all throughout your body. So I don't tell you all the other glands. This is just the way. The reason why you talk about this one controlling is it's known as the master gland, because there are actually 7 categories of hormones that come out of the anterior pituitary, and there's 2 that come out of the posterior pituitary, and so they are stimulating. The other ones can be due to nerve stimulation, due to feedback mechanisms, whether it's temperature or pH. Other hormones can stimulate it. It could have receptors that have low, they don't detect enough of something, let's say calcium or something, and so as a result more is generated. So any of your feedback mechanisms. Okay, what else? Yes, sir.
I have a question from chapter 38. Could you explain the process of sensation?
Sure. Okay, can I finish with this and then I can go back? Yeah, okay, go ahead.
For FSH and LH,
I cannot hear.
do we need to know like the specifics on how it works, or just
The reason I brought up the whole reproductive cycle, if you haven't learned the menstrual cycle yet, don't focus your time tonight learning the menstrual cycle, okay? Do I want you to know the menstrual cycle for your final and for the AP exam? Yes, I do. Okay, but if you haven't learned it tonight, I don't want you to spend 20 minutes trying to learn the menstrual cycle instead of reviewing some of the other things. Okay, I am telling you that much. And telling you that, I also want to tell you, when I was writing the test yesterday, I had like a list this long of stuff I needed to get done. I still haven't written my sub-plans. I'm not trying to whine about it, but I forgot to put in how the suggested reading and thinking. I do not know what's wrong with me, okay? But could I eventually ask you about those? Yes, so make sure you have them, okay? No, here's the eventually. You know where it's going to be? It's going to be from this semester, and then it'll tell you which ones to study. The ones I don't have test on, which is this test and the last test, expect those on your final. Okay?
Yes. For ACTH, would we have to know exactly how it stimulates the adrenal cortex? Like,
No.
No.
Just know that it stimulates the adrenal cortex. Okay, what else on the endocrine system? Feeling confident? No. Okay, so next let's talk about development. Okay, so what do you want to talk about with development? Yes.
Somites.
Okay, so somites are tissues along either side of the spinal cord that direct the development of that particular tissue for this particular segment of our body. So you need to know it as well as the definition is. Did you have it as one of your terms?
Yes.
Okay. What else? Uh-huh.
Can you go over the notochord and the adrenal plate?
Sure. Okay, so this would be a really good example of induction, right? So at the point where you're going to form the nerula, which would be the organ level, or system level, you're starting in on your nervous system. The notochord, which is from the mesoderm, induces the tissue above it to start to form this neural plate, this neural groove, and then a neural tube. And it basically is causing invagination of the ectoderm to form that dorsal hollow tubule nerve cord. And then the notochord for vertebrates will actually become the bones that are our vertebrae that protects our spinal cord. Is that what you want to know?
Yeah.
Okay. Yes.
Will we have to know like morphogenesis and cytoplasmic segregation?
Yes. So, but I would make it logical. It's logical, and I'll try to tell it to you as logically as I can off the top of my head, okay? So, first of all, there are differences within the cell maternally. As it's getting fertilized, we know that there are differences. There are differences in the amount of yolk, right? That gives you your dorsal ventral pole, right? So when you look at an egg, there's probably more yolk here, and you know this is going to be the ventral region, and this is going to be the dorsal region. And then wherever the sperm penetrates the egg, that's going to be the anterior region, and opposite of that is going to be the posterior region. So you have differences set up right away. Depending on the organism, the word is totipotency, where you have full developmental potential. For some organisms, the very first cleavage, the very first division, like for mollusks, okay, the very first division, that's it. Tissues are already determined as to what they're going to be. And if you separate them, neither one of them will develop normally. For us, we can get up to about 8 cells or so, maybe even 16 cells, and you could still space them all out and they would still have totipotency, full developmental potential. So it varies per organism. But the heart of the matter is there are differences within that, and at some point those differences are too specific and you can't separate out the cells and get any kind of normal development. So we have these regions right here. So when you start to do, when you look at this, you can see it as quadrants, and there are differences. And we talked about this. We said this has yolk and it has sperm. This only has yolk. This doesn't have any yolk or sperm, and this one only has sperm. So we could see differences in there. As the cell starts to undergo cleavage, then whatever is in the cytoplasm is going to be slightly different. What those things are morphogens. Morphogens influence morphogenesis, right? And that's because they're turning certain genes on at a specific time. And then when those genes are turned on, it's kind of like dominoes. You know, it's like that gene is turned on, then that turns on that gene, and that makes a product, and that turns on that gene, and that turns on a gene. And so it happens in sequence. Now, morphogenesis is the development of the different tissues, and they will rearrange in shape and location due to morphogens. When I influence your morphogenesis, what is that called? If I'm influencing your morphogenesis? Induction. What would I influence you with? Morphogens. What do those morphogens tend to be? Transcription factors. Right? And we know about that because transcription factors can assist what? RNA polymerase to bind to the promoter and turn on a certain set of genes. What are those genes called? Homeotic genes. Within those homeotic genes, there are things called homeobox genes. Just think a box is in the home. So the box is the most specific of all that's coding for those transcription factors. So I think we said all the different words, right? I said all the words you asked me about?
I think so.
Okay, what else? Like, I don't know, but that screwed me up. Okay, go ahead.
Do we need to know like the developmental process of babies, like week 1, week 2?
Just in general, I would note, I'm not asking you if you watched the essay, I said I wouldn't essay you it over it, but maybe you could know some general things that we talked about. Remember we said you would blast, remember this? Go through that like summary page, but I don't have an essay about it. You know, like week 4, what do you have?
Heart.
You have 4 limbs. It's 4th months when you can hear the heartbeat. 4 limbs and what? 5 senses. What week does the placenta develop? 10th week. Yeah. Blast into the 1st week. You are a blastula by the end of the 1st week. The 3rd week, you are a nerula, and your heart is starting to develop. Yeah. Okay, I saw a hand up there somewhere. Yes.
Oh, I have a question about Hox genes. So, do the genes make like maps on the organism? Like,
Well,
how does that
what
work?
happens is when you look at those Hox genes, the critical part there is the similarities amongst different organisms, that they have the same genes in the same sequence coding. You know, it doesn't matter whether you're a rat or a goat or a person, this Hox gene codes for the anterior region. How that plays it out, it's telling you anterior region develop. It's controlling that. It's inducing, right? Whatever it's producing is getting the anterior region to develop there. Okay, and then the next gene is going to get the next segment and so forth. And you see the similarities in genes among several different organisms. That's what I want you to walk away with on that one. You are welcome. What else? Okay, well, hang on to that thought. If something comes up, I'll come back to it, okay? You thought of one right now? Okay, go ahead.
Do we have to know like the gray crescent frog thing?
Why is the gray crescent? What is that a good example of?
Induction.
Well, at the gray crescent, we know that that area is important because that's where invagination occurs to form the gastrula. Okay, so that's what's significant about that gray crescent. If you forget what the gray crescent is, it shows up on the opposite side of sperm penetration. And when they had cells that didn't have a gray crescent, they wouldn't do the gastrula stage, because that is the dorsal lip of that blastopore, the opening, right, to the gastrula. Something else, development? I'll come back to it. I'll re-hit every one of those that you want me to re-hit, but I want to answer your, yes?
Sorry.
That's good.
Going back to induction, you were talking about induction with the morphogens, and
Yes.
then it also talks about the direct contact. So would that?
That would be another way. Yeah, use morphogens or direct contact. Absolutely. I thought in my head. I didn't say it out loud.
Wait, but what was an example of the direct?
I didn't give you one of those, so you don't have to worry about it. I want you to focus on. Yeah. It literally is one neighboring cell. Do you remember how we talked about prostaglandins, how they can affect a neighboring cell? It would be similar to that. Yeah. Okay. Anything more? Write it down if you want to. I'll come back to it. So you asked me about signal transduction, or what did you have? Sensation?
Yeah.
Okay. So in the process of sensation, what you want to get in that is, don't make it harder than it is, okay? But what are you going to need to sense something? What are you going to need? Oh, yeah, you'll need a stimulus, first off, and then you'll have to have some sort of receptor. That receptor may be a cell itself, or it may be an actual sensory neuron. Understand the difference? So for instance, in taste, this is just the simplest example. Taste, there's a taste cell that can trigger a taste neuron. But in smell, that's an olfactory cell. It's actually a neuron, a live action neuron. Okay? So once you have triggered that, okay, that's either a cell or directly a sensory neuron. That sensory neuron is going to go up in here to your proprioceptor. Eventually, it's going to connect with an inner neuron and then go to your brain. And you can have a stimulus that goes there, but it's secondary. You have to perceive what that means, right? And remember that picture I showed you? It had like a cut up person and a face folded, and you thought, oh, is that a real picture? No, it's a photo that's cut up. You know how you can be tricked? Your mind is tricked. And so its perception of something is different than perceiving it. Okay? Or wait, that didn't make much sense, did it? What do I want to say on that one? Your interpretation of the sense is how you're perceiving that, what that means, right? And so the meaning of the sense is different than just receiving that sense in your central nervous system. So what I'm trying to say, you have to have an awareness of what that sense means, and then give your appropriate response. That was it for that one. What else? Anything back on excretory system that you want to talk about? Feeling confident, the nephron, urine formation, good. Nervous system, you're good. You understand resting potential, action potential, recovery. Yes?
Yeah, actually on the nervous system, could you explain the undershoot part of the thing?
Yeah. What this is that it's important because you're changing the ion concentration. You want to make sure you're trying to, what our neurons do is transmit a message from point A to point B. And what happens is if you're opening up these sodium gates, and sodium is going in, it's bringing in what kind of charge to the inside? Positive, right? And that reversal of charge is going around along the length of the neuron, and it's immediately having a recovery. Well, what stops it at some point from moving backwards? You know what I mean? So you never get from A to B, you just kind of, and it fades away or something. You keep it going in one direction, and the reason is you overcorrect right after it happens. By having, what's the quick fix on a neuron? Potassium going out. And you actually have so much of that going out that it makes so much positive going out that what is it going to make the inside? Negative, really negative in that area. So that's the overcorrection. So it's easier for the neuron to keep traveling in that same direction you wanted it to go, because to go backwards, you would need a lot more stimulus in order to overcome that negativity. Yes?
What's the threshold that you want us to know? I know in some places it's like -50 and other places it's -55.
Yeah. either one is fine, as long as you mention the threshold. You don't have to know verbatim that one. Yes.
So the threshold, it's achieved because like a chemical message has to start it, right?
Okay, so your neuron, when you have your resting potential, okay, you can be stimulated, a little sodium come in, a little sodium can come in, but not enough to reach that threshold to trigger that neuron to fire. So that threshold is like go, no go. That's when you're deciding that. And it's once that is triggered, then you're definitely going to fire that neuron. The sodium gates are going to start to open and it's going to move forward. Thresholds can vary, right? Because we know the difference between warm and hot, right? There can be variations within that. We're able to have a very complex understanding of the stimuli in our body. It could be for one of two reasons, and we didn't go into great detail about this. It could be because various thresholds, or it could also be due to how many times you hit that one neuron with more stimuli. Yes?
For the long-term fix
For the neuron?
Yeah, the neuron.
That thing.
What is the long-term fix for?
Because the quick fix is, so well, you already know sodium came in. That was your action potential, right? All along. Sodium in. The fastest way to fix that is to have a replacement positive charge go back outside, so the outside becomes positive again and the inside becomes negative again, so this neuron is set to fire again if you want to use it again. So the quickest fix is to have potassium go out. Well, if you only did the quick fix again and again and again, pretty soon all the potassium would be outside and all the sodium would be inside. These gates work in one direction. So to cause depolarization, you need sodium to come in. Well, if all the sodium's in, you're never going to depolarize that neuron.
So the sodium potassium pumps
Puts everybody back in their places.
Okay.
Yeah.
But the potassium gates open no matter what, right?
No. The potassium gate is going to open, but these are one-way direction gates. So when the sodium gate opens, it can only move from the outside to the inside. Okay? That's the only way it can move. The only way you're going to get sodium back outside is using the sodium-potassium pump. So it's kind of like once the sodium comes in on that gate from the outside to the inside, it will stay inside of there until the sodium-potassium pump pushes it back. It can't, the only one is the potassium gate is kind of leaky. So sometimes potassium leaks back out, and that helps contribute to the resting potential.
Okay.
Yes?
With excitation and inhibition in
Mhm.
like the calcium like transition across the synapse, why would inhibition occur?
Well, for instance, I'm looking at something now. I'm looking at that door, right? And I'm perceiving the door now. When I look over here, I don't see the door anymore. Okay? So you have, they're called in your eye, okay? You have rods and you have cones, but then you have these bipolar ones, and they're inhibiting every neuron either side. So if I get stimulated, then I have this other neuron it's synapsing with that inhibits. And the reason why you do that is to accentuate edges. So that might be one thing that I want to do. Or I might want to inhibit it because I don't want to keep, have you heard of tetanus? Okay, so I want to contract a muscle, but once I contract a muscle, I don't want to sit here and just keep contracting it, right? I want to stop contracting that muscle, okay? So there's times you want to start and stop a stimulus. And if I go, hey, I don't want to go, hey, you know what I mean? I'm controlling my response to you, and it's a mixture of stay and go, right?
Okay.
Yeah.
That makes more sense.
Yes.
Would inhibition ever be like a conscious thing, or no? Is it just?
Yeah, for me, all the time. Right? You with your large mammalian brain, you're inhibiting the choices and the responses you make, right?
Okay.
You're not going to just stand up here and say something real rude to me because you would be like that. Even if you thought it, you know, you wouldn't stand up and shout it. You're inhibiting that response, right? You're making a choice inside your brain.
Okay.
Sometimes people don't have that, right? I'm sure we've all met people like that. Yes.
In the hypothalamus and the pituitary,
Yes.
the anterior pituitary, when the hypothalamus is sending like inhibition and
What do you mean the hypothalamus is sending inhibition? Speak.
isn't it like, doesn't the hypothalamus send hormones that
Okay,
are
so
secreted?
what happens is you will send, if you have, let's say, thyroid hormone,
Yeah.
thyroid hormone inhibits the release of thyroid releasing hormone and thyroid stimulating hormone. So the presence of it Okay, so you'll have receptors in both the hypothalamus and in the anterior pituitary for thyroid hormone. So if you are receiving thyroid hormone, then you won't secrete thyroid stimulating hormone or thyroid releasing hormone because you have, so that's how it controls it. And remember, you can have 10,000 synapses on a neuron. Remember we talked about integration? So it might have to hear 700 yes votes, right, and 300 no votes, and then it adds that up. It's got 1,000 votes total, but more yes votes than no votes. Do those yes nodes, throw it over the threshold to stimulate that neuron to fire. Yeah.
About the reflex arc, is that in the spinal cord or the somatic system?
Well, it's going to use motor neurons of what we consider the somatic nervous system. So remember we talked about it, like I can control if I know I'm touching a hot pan, right? But the reflex arc is just referring to the way our body protects ourselves. Just like we have internal reflexes to regulate our temperature and our pH and our heart rate and our blood pressure, we can have reflexes too that protect our body as well externally. So you are still going to use the nerves of the somatic nervous system through that part. Anything else? All right. Be awesome. I won't see you tomorrow, but I hope you do a great job. What do you have to have ready for class on Tuesday? Yeah, make sure you're good on that. All the way through the data tables, right? Okay.
I also have a quick question.
Yes.
Critical thinking questions.
Yes. Just let me pause this real quick. And don't stay up too late and have a piece of toast.